fine. Use your
Of course.
Thank you. So whenever you're ready, you can just click.
You got it.
Thank you so much.
No problem. There we go. Okay, that's good. You guys have never heard this before.
Nice.
I know. Okay. All right. Good. Okay. Good morning. Feel free to grab a snack or anything, and also interrupt me for questions or anything. But my name's Kara Davis. I am a clinical instructor in pediatric oncology in the lab. And this morning we're going to talk a little bit about some of the applications that we've used, mass cytometry and some of the single cell data for understanding normal human development, and then how we've applied that to looking at malignancies, specifically in this case, B-cell malignancies. So we're going to talk a little bit about leveraging high-dimensional single-cell data to model hematopoiesis in some healthy and malignant systems, and how we've developed tools to analyze and visualize that data. And you're going to hear a lot more in the next kind of set of talks this afternoon, or this morning actually, about some of the tools that have already been used and published on, such as SPADE, and the more specifics about how to use those, and then also some of the things in development. And so I'll refer to some of those things, but I'm not going to go into great detail into all of the different particulars of the algorithms, but we can always talk about that after if people have specific questions. So I think most people can agree that developing cells, when you're looking at them by flow, they can be linked by phenotype. This is a nice review from 2004 that was demonstrating the progression of, in this case, is this the pointer? B-cell development based on CD20 by CD10, that you can see that cells kind of follow this line of development where they initially have CD10 and then lose it as they gain CD20, and that you can track them through development in that way. And you can do this in most lineages if you know the sufficient markers to look at, including myelocytes, monocytes, and erythroid cells over here. But the difficulty with this kind of looking at this in lower dimensions or with fluorescence cytometry is that it can be difficult to decide where to draw a gate on one of these two-dimensional plots. And so you may not be able to capture all the cell types sufficiently with either the markers that are chosen or with the gate. Although we've come pretty far in identifying cell types just with this level of detail. And one of the problems is that during development, there are some relationships that are non-linear. So if developing cells, if they actually go like this along kind of a curved line, that if you use something like PCA to try to fit that trajectory, you're not actually going to capture the direction of development. So you're going to miss things. And it doesn't matter how hard you hammer at the PCA component, you're never going to be able to actually get this kind of trajectory out of the data. So we were working with Donna Pear's lab at Columbia with a grad student named Elad Amir, who was working on trying to develop tools to better determine trajectories in nonlinear data. And so what we came up with was an algorithm that's called Wonderlust. And basically how Wonderlust works is that you have your n-dimensional plot. So in this case, we had normal human bone marrow and about 40 dimensions, a little more than 40 dimensions. And so an n-dimensional plot, there was a 40-dimensional plot of single cells that had some shape in high dimensions. And the first step in the analysis is to create a nearest neighbor graph so that every cell gets connected to some amount of nearest neighbors. So let's say 5 nearest neighbors. It's 5 nearest cells in high dimensional space. And then you would do this over and over again. So you would make a whole ensemble of nearest neighbor graphs. And the idea then was to try to determine the shortest path through the graph from a user-defined starting cell, which is red here, to for any of the other cells in the data set. And this was done by also giving the data set landmarks, randomly chosen landmarks along the way. And so then it became, I like to think of it kind of as a triangulation problem. So if I'm trying to understand where cell X lies in my data set, you would determine the shortest distance from cell X to the starting cell and from cell X to the landmarks. And this process would happen over and over for every single cell across every single nearest neighbor graph until you came to a kind of an agreement of where is the position of every cell in your data set. And these were then averaged across all the graphs until you eventually end up with something that looks like this. So parameter A, let's say, across the Wonderlust axis being X, would be a parameter that the cells were low at the beginning and then rose and then decreased. Versus parameter B might be something where cells were high at the beginning and the level of that marker decreased across the entire data set. So we actually applied this to look at B cell development because we thought that B cells could be a good model because they're generally linear in their development, so we think, whereas myeloid development or erythroid development is more branching probably from the stem cell. And what we found is that our predicted trajectory matched a lot of the known facets of B cell development. And this is without really any manipulation of the data up front. So cells were basically gated out for non-B cells or lineage cells, T cells and myeloid cells, and then the algorithm was run. And so the axis down here, which we call the Wonderlust axis, can also be thought of as kind of developmental time. So 0 being the earliest cells and 1 being the most mature cells in this data set, which would be any kind of a B cell that is still residing in the bone marrow. And what we found was that, as we expected, CD34 came first, followed by CD38. Next was CD10, and then CD19, CD20, and immunoglobulin heavy chain. And then, whoops, oh, I didn't have them fly up, but kappa and lambda come last, the light chain components, which is exactly what you would expect for how B cells would gain these markers. And the really nice thing was that the predicted trajectory was bidirectional. So if you if we told the algorithm, I want you to start at a HSC or a CD34 positive cell versus a mature B cell, it can find the trajectory by going either direction. So it was able to find the trajectory from the beginning or from the end, which is nice, I think, for the thought of how you could use this for systems in which you may not know what the beginning cell is, but you know what the mature cell is. And there's many more instances in biology where we know the mature cell, but we don't know the progenitor cell. So this higher dimensionality allowed us to kind of uncover populations that had not previously been described in the human. So what we noticed on our trajectory was 34 coming up and 38 coming up, understandably. And then TDT, which is the enzyme that helps add bases to immunoglobulins when they're being rearranged, was the next kind of earliest marker that we thought indicated a lymphoid type cell. But then what we noticed on this red trace was that this marker CD24 came on much earlier than, not much earlier, but earlier than other B cell markers like CD10, which we kind of expected to be the earliest marker that we would see that would mark B cells. So when we looked then at the Wonderlust values, actually, wait, I'll go this way. So if we went back to just our biaxial data and looked at CD34 by CD38 and first identified our stem cells down here, if we went into the double positive population here of progenitors, if we gated based on these two markers in 2 dimensions, we could see that there were 4 nicely gateable populations there, which had not really been previously described in human B cell development. So we labeled these population, so HSCs being population 1, population 2, 3, 4, 5. And when we looked at the median Wonderlust values of these 4 populations, it appeared, or actually these 5, it appeared that they were gaining maturity. So that cells, as you would expect, would start in this compartment. They would move into this double negative compartment, then they'd gain TDT, they'd gain 24, and then they'd lose TDT as they were going through development, as indicated by their rising Wonderlust values. And when we looked at other markers, because then we had many more markers to look at, when we looked at other marker expression on these different cell populations, what we found was that if you looked at expression of lambda 5 and V pre-B, which are components of the early surrogate light chain of the pre-B cell receptor, that initially in the stem cell fraction and in the progenitors, you start to see a little bit of lambda 5 coming on, but then there's co-expression as they move to fraction 3 and 4, and then they start to turn off by the time they get to fraction 5. And then when we look at CD10 or immunoglobulin heavy chain, we see a little bit of expression of CD10 by the time they're here in population 3, but it really isn't kind of maximally on until population 4, and then it's lost by the time they get here to population 5 as they're expressing heavy chain. So we felt pretty confident that these were probably early B cell precursors, but the way to test or to tell was that we sorted them out in 2 additional bone marrows, and then did an RT-PCR assay to see if they actually did have rearrangement of their heavy and light chains, which would be kind of the gold standard of deciding if you were a B cell and how you were progressing on your clock of being a B cell. And what we did find was that as we started here from population 2 and moved around to population 5, that there was progressive gain of DJ rearrangements and then also VDJ rearrangements. And if you're not a B cell person, it happens in that order. You do DJ rearrangement first and then VDJ rearrangement. And by the time cells got to population 5, most of them were rearranged, had a VDJ rearrangement, and we also knew that on the protein level, a lot of them were expressing immunoglobulin intracellularly. And just to kind of give you a sense of how rare these cells were, the cells in population 3, so if we take the slice of all the mononuclear cells in the bone marrow, this fraction, the CD34 positive 38 positive fraction, was this red slice right here. And then if we kind of take that slice and divide it into another pie chart, you can see the relative proportions of these different populations. So this kind of mixed progenitor population 2 was the most frequent population there. And then population 3 was the most rare, being 0.007% of cells in the marrow. So it was really very rare. But the fact that we were able to look at single cell data and collected sufficient amounts of events that we saw this population kind of in our very first runs on these normal bone marrows, and then when we went back and did more of them, knowing how rare these were, we kind of modified our experimental process to try to get more of these cells by starting with more marrow and depleting lineage cells at the beginning. So again, since we were able to look at many, many markers, and also a la Nolan style, we did stimulations for these cell populations as well. What we also found was that as cells went across these from population 2 to 4, they also began to express the IL-7 receptor. And if we stimulated them with IL-7, by the time they got to population 3, where they really start to turn that on, you can see a STAT5 response when you stimulate them with IL-7. And if we look at this across different biological replicates, so different healthy bone marrows, and this is all normalized to the basal control, that there is the highest induction of STAT5 in this population 3 when they're exposed to IL-7. And then further, we could look at what else can stimulate STAT5 in these populations. So not only IL-7, but also TSLP, which is thymic stromal lymphoprotein, which is another known kind of cytokine that can stimulate IL-7 in lymphocytes and seems to be important in allergy and atopic disease. So in these early populations, and TSLP actually uses the IL receptor alpha chain as part of its receptor with its own TSLP receptor. We could see that if we treated these cells with a JAK inhibitor, tofacitinib, which is a mostly JAK1 and 3 inhibitor. In addition to IL-7, we could abrogate some of this STAT5 response to IL-7, but that we could still, across all the populations, we could still get a STAT5 response if we treated the cells with pervanidate, which is a phosphatase inhibitor. So it suggested that across these early populations, there was a rewiring of at least the IL-7 STAT5 signaling network that was happening across development in these really rare populations. And if we go back now and kind of zoom back out and look at this across Wanderlust, this shows up here kind of labeled generally where those fractions lie, and a reminder that right in between population 3 and 4 is when we think that the IgH locus was rearranging. And we can see again that the IL-7 in red here is IL-7 receptors coming on, and TDT is mirroring that, but then kind of peaking beforehand. And so this bar down here is colored by basal STAT5. And what we again noticed is that there's kind of low level, not very much STAT5. And even in population 3, which is the most inducible in response to IL-7, there's not a lot of basal STAT5. But by the time you get to this population 4 to 5 transition, the cells have high basal STAT5, which could not really be further induced by IL-7 across all those different samples. So it seemed like what was happening as cells were moving from population 3 to population 4 is that when they're in population 3, they express the IL-7 receptor. They can respond to IL-7 in a JAK-dependent manner to activate STAT5. But as they transition to population 4, they no longer are responding to IL-7, regardless of the fact that they still express the IL-7 receptor. But they then still have kind of their STAT5 signal turned on. So either if that's by inhibition of a negative regulator of STATs or some other mechanism feeding into STAT signaling, we were not quite sure. But that does suggest that there's actually kind of a remodel of the system that's happening between these two stages. So taking a really great idea from Tiffany Chen, who was probably here yesterday or will be today talking about Cytobank, she suggested that we take a look at what's happening and how all of the markers that we measured are moving together across Wanderlust to see if we could find some sort of coordination in how the proteins are moving. So what we did is we took our Wanderlust signal, we kind of sliced up the time across into analysis windows, and we took the first derivative to then get a heat map that looks like this. Oh, nope, looks like that. In which blue would indicate a negative slope, meaning that a marker is decreasing in its expression, and red would indicate a positive slope, meaning that a marker was increasing its expression. And again, if we go across time here, we can see that there are some patterns that kind of emerge. And here in the first red box, we see this first kind of coordination point. And this is up here as well. You can look at it on the Wanderlust trace. But essentially, we see that there's a dramatic rise in CD24 at the same time that the c-KIT receptor is decreasing. There's this rise in the expression of the pre-B cell receptor components, and also CD127, the IL-7 receptor. And at the same time, TDT is also rising. The next checkpoint that we noticed came shortly thereafter, and that's in this Wanderlust trace over here, right about 0.4 on the Wanderlust axis. And what we saw is that's about the time that CD34 really decreased, TDT is decreasing, and there's a rise of a lot of other kind of more B-cell-centric markers like CD19, CD72, CD20, and we start to see PAX5 come on, which is another B-cell specific transcription factor, and that's also when the pre-B cell receptor components are turning off. So this is right about population 4 to 5 in our early populations. And as we get out later, around here, we see CD20 coming on at about the same time, or CD20, sorry. CD20 coming on at about the same time as kappa. And then a little bit further out, this black box, we see other markers of more mature, naive B cells that are ready to leave the bone marrow and go out into the periphery. So IgD, CD40, CD22, and CD38 declining. So we started to be able to kind of pick out across this wide, I guess, look at development across B-cell development, these checkpoints or alleged kind of checkpoints where things are changing, at least at the protein level, kind of in concert across development. So taking then another look at some of the other markers that were moving at these same times, we wanted to take a look at, we only had a few markers kind of cell cycle, not to the same level that Greg had, because we didn't have the ability to kind of get to these bone marrow samples and IDU treat them right out of the patient, or the donor, I should say. But we did include cleaved PARP, and we did include Ki67 to try to pick up cycling cells. So what we noticed is that this heat map here, the background of it is colored by Ki67 expression, so white meaning high Ki67 or proliferation, and the dark gray or black meaning low Ki67 expression. So right here, where these cells, population 3, are responsive to IL-7 and STAT5, it seems to be in a timing of increased proliferation for these cells. And soon after, we see rise of the surrogate light chain components V'(B) and lambda-5, but no expression of heavy chain yet. And as they move to population 4 and they're rearranging their loci, this is when they become, they're not cycling, so they stop cycling, which makes sense because you probably don't want to be cycling when you're cutting up your DNA for immunoglobulin rearrangement. And just before they go back into cycle, and just before they then start to express a heavy chain, we see this spike in cleaved PARP, which we think seems to correspond probably with cells who are undergoing deletion for a non-productive IgH rearrangement. So going back and kind of tying this into those coordinated protein markers that we saw in the previous slide, we think that these are calling out these pro-B cell checkpoint and pre-B cell checkpoint, which are pretty better understood, I'll say, in the mouse than in the human. And so being able to kind of tie this together with some functional cell decisions about when to proliferate or when to not proliferate. So we wanted to kind of look at this in another way. So we did a cell culture experiment, a co-culture experiment. We took 2 other healthy human bone marrows, lineage depleted it, and for 6 weeks it was plated on OP9 stromal cells, which are known to support B cell development, plus or minus different kinase inhibitors, and then went back just by fluorescence cytometry after that 6 weeks to see what did we end up with. And the result showed that, as we would have expected, that for the, let me make sure, so everything's normalized to the DMSO control here, which is black, that the 2 JAK inhibitors that we used, ruxolitinib, which is in blue, and tafasitamab, which is in red, that in those 2 cases we saw that there was a pileup, basically, of cells in population 2 and less cells that had progressed through to population 4, suggesting that that blockade of the JAK and STAT5 signaling was not allowing as many cells to progress across this developmental transition, or this, what we think is probably the pro-B checkpoint. And just another interesting observation, we used a P38 inhibitor and noticed that that is the green, that across the populations there was actually almost an increase, especially in these later populations, in the number of cells in those conditions, suggesting that the P38 inhibitor was kind of permissive to a non-compartmental increase in cells. Dave.
Were you able to track the progression, or did you just sample everything at 6 weeks?
We sampled everything at 6 weeks. Yeah, we didn't track the progression. And actually at 6 weeks, we kind of thought 6 weeks was going to be long enough to get more cells in the later, like even out to population 5 and 6, but it wasn't. Like we probably could have gone longer than 6 weeks. So basically what we ended up with was this kind of summary of what's happening in the human system in early B cell development, which is something that hasn't been well delineated, mostly because these cells are really hard to get. People usually do this work in mice, and so, but there are known to be differences between mouse and human, and so we think that we've done a pretty good job in calling out some of these important stages in early B cell development that will be helpful to further investigate in disease states. So if we compare them to kind of what's reported in the literature and how this lines up with our fractions, we can start to kind of call out what are the sufficient markers to sort these cells if you wanted to sort them on a fax, and also the expression of other kind of not bystander markers, but markers that are also there at the same time on these fractions. But we can start to see, again, how protein expression is changing across development. And then additionally, we can line it up with some of the regulatory signaling and cell fate decisions that are happening across these cells. So where the IL-7 STAT5 response is, where there's high basal STAT5, when they're responsive to B cell receptor cross-linking, which I didn't show you guys that. and then when they're having a DJ and VDJ rearrangement. And just to remind you that this all happened in very, very few cells. All this stuff that's happening in the beginning is not happening in a lot of cells. But because we had single cell data, we were able to kind of capture these populations. So quickly, in my last 5 minutes, I want to talk to you a little bit about how this then has kind of laid the foundation for my looking at leukemia, or B-cell leukemia. And I'm highlighting kind of this box here that it seems to be, now that we know what's happening in these early fractions, that, and not surprisingly, that B-cell acute lymphoblastic leukemia is arising from populations that kind of are sitting in this checkpoint, these checkpoint areas, and so suggesting that this is really a time where things can go wrong and lead to malignancy. So we had, in the initial kind of experiment, 9 pediatric pH-positive ALL samples. So these are all characterized by a specific genetic translocation. They'd all be treated the same way in the clinic with the same kind of medications. And traditionally, these patients have pretty poor outcomes. They don't do very well. And then we compared it with healthy bone marrows. We looked at a pretty extensive panel of markers that had mostly an overlap with what we did with the healthy B cells, and looked at it in a couple different conditions with inhibitors. And so the question I really had, and what I came into this like backwards, I was interested in the ALL, but then we did the B cell stuff, and now it's like going back to the ALL, but it worked out well because it kind of educated my approach to the ALL. But typically, as a clinician, like when I have a patient that comes in with ALL, they get diagnostic flow, I get the report, and what do I do with it? I see in the blast gate, oh, they express 34, they express 19, they express 10, or TDT, and I can say, okay, this looks like B-cell precursor ALL, you're going to get this therapy, and I don't really ever think about the flow again unless they relapse. But the question really is there more information hidden there? So if we go back and look at the biaxial plots from like what you may get from a clinical flow lab, you can already start to see between these two patients that there is some differences. So they both highly express CD10 and CD34, but you can start to see some differences in the expression in CD19 and CD20, and especially compared to a healthy bone marrow, they're certainly different. So when I initially approached this data set and just going straight to SPADE, if I drew a SPADE tree using all of the data from all of this set, the two healthies and all the leukemias, you can see that this is the tree structure. And in the healthies, almost all the cells localize over here on the left side of the tree. So there's two healthies. As we go through them, they kind of overlay right on each other. But as you move to the leukemia, they are expanding this side of the tree. But every patient kind of occupies this part of the tree really differently, although there's definitely some areas that are shared or kind of in common to all of them, which is not something I really expected because I thought this was a pretty, these were all the same gross genetic lesion I expect, and usually when I was thinking about looking at flow, it all looked pretty much the same. So when we started to look into this a little more deeply, it seemed that this developmental trajectory was still applying to these leukemia samples, and especially even in this small kind of specific subset. So this is a different visualization that I don't know you'll necessarily hear specifically about, but it's published. It's VISNI, but in this case, these are not single cells, they're clusters. So these are clusters of leukemia cells visualized by VISNI. And so basically what we saw is if we took, this is colored by patient sample, that the leukemia kind of spread along this long kind of curved, I think of it as like an elbow, of the map. Whereas these cells down here are actually all healthy cells. These are T cells, healthy B cells are up here, myeloid cells are here. And if we start to look at marker expression, this doesn't project so great, but we can start to see that all the leukemia cells have 34 expression, although there is some gradient there. The cells up here in this top right-hand side are kind of the highest for 34 expression. And then there's also a gradient of 38 expression down here where the cells at the bottom part of that elbow are highest for CD38. And there continues to be some remembrance of the rules kind of normal B cell development where we see vPre-B and lambda5 co-expressed in the populations that also have CD38, which is exactly what you would expect in the healthy B cells. And if we looked at these populations again in all these different markers, if we cluster them based on phenotype, we kind of get three maturity groups, where CD34 is the highest and then kind of declines but never like goes back down. Again, we see this kind of hump in V-pre-B and lambda 5. They all highly express CD10. CD24 is low initially and then kind of rises, just like we expected in healthy development. And then again, TDT is low and then rises, and CD20 starts out low and then rises toward the end, so that these populations in maturity, or these groups in maturity, would go from this way to this, from 1 to 5. And this is a different way of looking at it just on the map, so that cells that are up here in this top corner are kind of the most immature looking, and then they move into this population here as like a pre-pro B cell. Then these cells here would be like your pro B cells, pre-B cells, and then an immature B, kind of like that transitional cell. And then you can look at, in every single patient, how do their cells occupy this particular, all these different phenotype groups or maturity groups. And just to give a couple examples, you can see across all of them that, let's say for the first sample, SJ01, that most of the cells are occupying this kind of B progenitor-like population, and then there's some healthy populations in there. But this patient also has some cells, about 5%, that are in this pro-B-like phenotype, whereas this person or this patient, most of the cells are in a pre-B-like stage, but they have a little bit of cells that are in a pro-B-like and a little bit of cells that are in a pre-pro-B-like. Most of the patients have kind of the majority of their cells in one particular silo. This patient was a little bit different, where they had kind of bigger chunks of the leukemia that spread across two different types of phenotype. And when then we looked at some of the signaling, basal signaling, you could see that it also recalled maturity stage. So this is looking at STAT5 on the map. And in maturity group 3, which is like our pro-B cell, and maturity group 4, which is like our pre-B cell, we see the highest levels of STAT5, which is exactly what we would expect in the healthy B cells. That's about the same time when they're having high levels of STAT5 basally and in response to IL-7. And the same thing with Ki67. The cells that are in this elbow here are also the ones that have the highest level of kind of cycling cells. So they're continuing to follow some of the patterns that we see in the healthy B cells at about the same maturity stages. So I'm going to kind of go through this quickly just for time. So we looked then at how could we, if we clustered the cells based on their response to dasatinib, what did we see? And we basically found that there were 5 different types of response to dasatinib. Dasatinib is a kinase inhibitor that you would potentially treat patients with this disease with. And one of the ways that people look at is dasatinib working is it inhibiting STAT5? And so in this case, we have 3 kind of types of response in which STAT5, which is this dark blue, is inhibited. And then we have 2 types of dasatinib response where you actually have some small induction of STAT5. And when we look at how does this compare to their maturity stage, what we see is that there seems to be a relationship between dasatinib response and maturity such that immature patients, or patients that kind of are, most of their cells look more immature, are more likely to be in these responsive types of response, whereas patients who have a more mature type of phenotype are more likely to have this resistance based on STAT5 signaling. And then these pies here are just to show that there's also, in every patient, not all of the cells fall into the same signaling response. So as a clinician, this is the kind of thing that could make you a little bit nervous, because if you think that you're treating a patient with a drug that should be working on their cells, you would hope that it's working on all their cells. But in this case, you know, at least based on this one way of looking at it, it's unclear whether or not a patient up here is actually going to respond optimally to the drug. So the kind of the future directions and how does this have me thinking about ALL is that, you know, thinking about what is the meaning of the developmental state and how does that impact a patient's response to a drug or to just the overall treatment in leukemia. So we kind of know now that the healthy developmental state across B cell development can be characterized by certain protein phenotype, signaling phenotype, and then that also likely goes with transcriptional profile or epigenetic profile and all of the levels of regulation in the cell. And so what I'm interested in looking at is then, if a cell is transformed into a malignant cell, is the position of where it's transformed meaningful in what is the outcome, such that a cell potentially that's transformed, let's say, in either these kind of progenitor or pre-pro B cell states in which that they may go into remission with treatment, whereas potentially if a cell is transformed in what's normally kind of a quiescent state, if that patient is more likely to relapse because their cells may be less kind of set up to respond to cell cycle directed chemotherapy, which is generally what we use in these patients. So with that, I'll finish up and just say thanks to Gary, of course, for all his support. And then the important kind of people to help with this stuff was Sean Bendel, who you heard from yesterday, Aaron Simons, Estrella is a huge help, and Angelica. A lot of computational help from folks, and as well as our collaborators at Columbia. So I'm happy to take questions. No questions? Zena?
How were the clusters generated with the classes?
So they were generated with k-nearest neighbor clustering and then community detection. So we can talk about that more kind of after the details.
Do you think it would be possible to look at like a personalized medicine approach, to look at like your signaling changes and then come in with like a SIG inhibitor, B2P inhibitor after looking at primary response?
I think you could, like in primary samples,
Patient.
yeah, that's what I mean, or in patient samples. I think it's possible if you had sufficient, number of samples. That's always like the limitation when you're working with primary samples, is that you don't have enough material. But certainly it would be possible. And I've done some more stuff recently where we did some patients on therapy samples, and just kind of as a pilot to see if we could actually get any cells from the peripheral blood of these patients. And so at least it looked like even as their leukemia was under treatment, we were still able to, early time, we were able to pick up some leukemia cells and be able to look at least from a phenotype, we didn't do any signaling, but look at the phenotype and how those cells are kind of changing.
Can you grow them in culture long enough to predict what the invasion?
So I haven't grown them in culture. ALL cells, from what I understand, are pretty notoriously difficult to grow in culture. I've actually, that's not true. I tried to do a couple things just with like prednisone, which is a staple of ALL therapy, and basically they died at the same rate if they were being treated or not treated. And so I think there have been, Dario Campana from St. Jude had a paper about ALL culturing, and it was kind of complicated as far as getting it to work. Not for those. Not for those. So those initial patients were from St. Jude, and I don't have the outcome data, but I just got 2 weeks ago like 50 samples with that all have 3-year outcome data. So that's the next thing, bigger sample set. Anybody else? All right, I think it's a coffee break, right? Let me double check it's a coffee break.
You gave us a coffee break.
Oh, no, wait, no. Oh, yes, it is. Oh, no, it's Eli. Are you next? I'm sorry, I thought it was coffee. Okay. Never mind, not coffee break. I thought it was 9:30.